Well, thank you, Lori, for that overkind invitation, and I'd like to thank the organizers for giving me the opportunity to talk with you this morning. I'm going to talk about sensations, and as I'll try and emphasize, the principal sensations that arise from the viscera are unwanted sensations, usually those of discomfort or pain. So the question is, what's the problem? And the problem is that there's too much feeling. Most of us understand that when your bladder begins to fill or your colon begins to fill, there's an appropriate signal to void. And that is a normal sensation and a normal feeling. But most of the time, you're totally unaware of what's happening in the viscera. You've had a cup of coffee, maybe you've had a muffin this morning. That's initiated a whole bunch of neural activity to initiate motility, the secretion of digestive enzymes, things of that nature, of which you're totally unaware. So the viscera sort of function automatically for the most part. Visceral pain is a problem, and the problem is that it's very different from cutaneous pain, and I'll try and emphasize that for you. And what underlies chronic visceral pain? There's a number of issues that we're going to talk about. I'm going to talk about how the sensory nerves, the afferents, are persistently sensitized, how one organ influences the sensitivity of an adjacent or a nearby organ, and that's called cross-organ sensitization. And then at the end, we'll acknowledge the fact that there's altered brain modulation that likely also contributes to these problems. So let's just look at pain as a topic and ask some questions with respect to the viscera and with respect to skin or cutaneous structures. You can ask questions, is injury required to produce the sensation of pain? Is the pain well localized, or is it reflected or referred elsewhere? And does hypersensitivity develop? With respect to whether injury is required, it's well known that injury or the threat of damage to tissue is important for cutaneous pain. This was developed, and our vocabulary about pain was developed by Sherrington in the late 1800s and early 1900s. And that has persisted, those definitions have persisted. But we've known for more than 100 years that you don't require injury to be present for visceral pain or visceral discomfort to develop. And the reason is that the stimuli that produce pain from the internal organs are different than produce pain from cutaneous structures. Excuse me. I can cut or burn or pinch the skin, and that's going to be painful. And I can cut or burn or pinch the viscera, and that is often without sensation. And so the adequate stimulus or the appropriate stimulus is different. And in the viscera, it happens to be distension or overfilling, traction on the mesentery, and then what we might call chemical stimuli like inflammatory events or ischemic events that influence the sensitivity of neurons that innervate the viscera. So the stimuli that produce pain are very different for those organs and skin, and that's true for muscle, for joints, and bone as well. The adequate stimulus is different to produce pain in those different tissues. And then you can ask other questions. What about localization? Well, localization of input from the skin is exquisite. I can blindfold you and prick your finger, and you can tell me not only which hand it's on, but which finger it occurred and where on the finger it happened. In the viscera, that's very, very difficult. And hypersensitivity obviously develops on skin. If you've ever had a sunburn, you know that you become hypersensitive during the period of that sunburn until it resolves. In the viscera, localization is poor and referral is common. Most people who have myocardial events don't reach for the substernal area, but the pain tends to be reflected up to the shoulder, sometimes into the jaw and down the arm. And hypersensitivity also develops. So it's key to remember that with visceral pain, the pain is not necessarily felt at the source, but it's referred to a nearby structure or a distant structure, and that hypersensitivity develops both in the organ and in the other areas to which the sensation is referred. So for example, people who have repeated myocardial ischemic events were established by a physician named Sturge in the 1800s to have hypersensitivity of the skin of the shoulder to which the pain was referred. And there's a neurological explanation for that, but you have to understand that when you palpate the abdomen of a patient who's got a bladder or a colon problem, they have hypersensitivity of the area that you're palpating, and that's related to the hypersensitivity that was established in the organ previously. So these chronic pains from the viscera then become a problem, and they're very difficult to manage. And there's reasons why they're difficult to manage, and the simple explanation is because we don't understand the mechanism. Well, we don't understand the mechanism because the viscera are complicated, and there are several contributing factors or reasons why it's difficult to establish mechanisms for pain arising from the internal organs. First of all, most visceral input is not consciously perceived. We talked about that a little bit. The principal conscious sensations are discomfort and pain. The visceral innervation is unique, and I'll show you some slides or cartoons about this in a moment, but all of the organs are innervated by two sets of nerves, and they have different functions, and you have to understand and appreciate that when you begin to address a problem of how do I control this person's pain problem. Localization is poor, referral is common, and chronic visceral pain, irritable bowel syndrome, bladder pain syndrome, dysmenorrhea, prostatitis, chronic pelvic pain syndrome is often characterized by hypersensitivity that's established in the organ, and oftentimes this hypersensitivity and this pain arises in the absence of a pathobiological, or at least an obvious pathobiological reason. So we have a lot to learn about how the viscera signal pain, and it's become a big problem, of course, for gastroenterologists, gynecologists who see patients, and other health science professionals who have a patient that's complaining about a visceral pain problem, which is becoming very difficult to manage. So here's a cartoon of the gastrointestinal tract on the left with the bladder and the uterus and the prostate in this unusual individual illustrated on the slide. What's illustrated on the right-hand side are slices from the spinal cord, from the thoracic to the sacral spinal cord, and then a section through the brainstem at the top. And the innervation of these organs is complicated. So the colon, the bladder, the uterus, and the prostate are all innervated, and by that I mean they have sensory neurons that are associated with conveying information from the organ to the spinal cord and then to the brain. They're innervated through nerves that form the pelvic plexus, and that's a complicated structure that has nerves wrapping around organs, and it's illustrated here as a simple circle, but the pelvic plexus is quite complicated, and that information is conveyed into the sacral spinal cord by the pelvic nerve, or also called the pelvic splanchnic nerve. Now that terminology is kind of difficult because there are many, many splanchnic nerves, and people tend to think of those as associated with the sympathetic nervous system. But the pelvic nerve is associated with the parasympathetic outflow from the spinal cord. A hypogastric nerve and a hypogastric plexus connects the pelvic plexus with the inferior mesenteric ganglion to form input to the thoracic spinal cord by the lumbar splanchnic nerve. Now, the lumbar splanchnic nerve is associated with sympathetic outflow. So many people talk about the sympathetic innervation of the viscera or the parasympathetic innervation. That's difficult terminology because the parasympathetic and sympathetic divisions of the autonomic nervous system are efferent systems. They control motor function, they control other functions, but they don't have sensory function. The sensory neurons are bringing information from the viscera to the central nervous system. And just to complete the picture and to complete the complexity, the vagus nerve is probably the largest furthest reaching nerve in the body. It innervates all of the organs in the thoracic cavity, which aren't shown here, and most of the organs in the abdominal cavity, perhaps even the bladder and the distal colon. So the key message here, aside from the diagram, is that each organ is innervated by two nerves, and they have different functions, and I'll emphasize that a little bit later as we go on. Localization is poor. There is viscerosomatic and visceral convergence onto neurons in the central nervous system. So here's a cartoon of a person, and it has a nerve ending in the distal colon with a cell body in the dorsal root ganglion, and it terminates on that neuron in the spinal cord, that red dot that you see there. And from there, that information is conveyed to the brain by a couple of pathways. But what's confusing about interpretation of that input from the colon is that that same neuron in the spinal cord receives input from the skin and the subcutaneous structures of the abdominal area. So the same neuron now is receiving two inputs, and they're confusing or conflicting in terms of how do I interpret this input. So that's viscerosomatic convergence. And almost every neuron in the sacral spinal cord receives input from another structure like muscle or skin or another organ. And so that is visceral convergence. So here you see that input from the distal colon that goes to the neuron in the spinal cord. In addition to receiving input from the skin, it's receiving an input from the urinary bladder or the prostate or the uterus. And so this neuron in the spinal cord is easy to understand that it gets a bit confused because it's receiving visceral and visceral somatic input. So if you have a bladder inflammation, a patient with interstitial cystitis, that neuron in the spinal cord changes its excitability because of the persistent input from the bladder. And because its excitability is increased, that means input from the skin or input from the colon is now also perceived incorrectly. So these inputs from the internal organs that have convergent input from other organs into the spinal cord contributes to this visceral convergence and cross-organ sensitization. And I'll show you a little bit more of that later. So what's shown here then are these sensory or primary afferent neurons. Their cell bodies are in the dorsal root ganglion. There's convergence onto a single neuron in the spinal cord. The information is conveyed to the brain by the spinothalamic tract, which is the traditional, conventional pathway by which information gets to the brain. But for the viscera, there's also a dorsal column medial lemniscal pathway that conveys visceral pain to the brain. So there's studies from the 1980s and 1990s for people with terminal colonic and bladder cancer that have pain that is relieved by midline myelotomies in the spinal cord interrupting this dorsal column medial lemniscal pathway. So there's definitely two pathways that bring information from the internal organs up to the brain. So cross-organ sensitization arises from visceral convergence onto the same second-order neuron in the spinal cord, but it may also be contributed to by multi-organ innervation from the same cell body. And I'll show you something at the end of the talk that how this might contribute. So the first evidence of which I was aware that the viscera exhibited this hypersensitivity was a paper by James Ritchie in the 1970s. And what he did is that he put a balloon into the colon of patients and distended it. And you can see in normal subjects, those people responded by saying it was painful at about 60 mils of air. And then as the intensity increases, of course, the sensation increases. But you see in IBS patients, that same distending volume produced the sensation of pain in almost 60% of the patients. So this is a leftward shift in the psychophysical function. This represents an increase in the response magnitude and a decrease in the response threshold. This is similar for interstitial cystitis patients. This is work from Tim Ness. Healthy subjects versus IC patients, looking at both intensity of pain and the unpleasantness that they report. Again, there's this leftward shift in the psychophysical function. So hypersensitivity is expressed, and in fact it's operationally defined as a leftward shift in the stimulus response or the psychophysical function. And hypersensitivity, whether it happens in the organ, in the spinal cord, or in the brain, really reflects, quite simply, a change, an increase in neuronal excitability. So the question is, what sustains these chronic pain states? So Clive Wilder-Smith published a paper in 2011 looking at potential top-down sensitizers, that is, the influence of the brain on these events, and the influence of peripheral organ-related events contributing. And these bottom-up sensitizers include sensitization, inflammation, and factors in the lumen of these organs, whereas top-down sensitizers are associated with catastrophizing and hypervigilance. I have to say that the top-down sensitizers have become very, very important in the literature these days because many people are doing brain imaging to look at what happens in these IC patients or these IBS patients. But they don't explain mechanism. On the other hand, if you look at the bottom-up sensitizers, you can at least begin to interrogate mechanisms at the level of the organ that are associated with these chronic pain syndromes. And so we're going to talk a little bit about how you go about looking at some of these things. And the easy way to do this is to record from those sensory neurons that innervate the bladder or the colon or the prostate or whatever your organ of interest is. And so you can take out the bladder or any one of those tissues, put it in a dish, and then, excuse me, and then you can probe the organ by stretching it, by pulling on it, by probing it, and you can identify where the receptive ending of the neuron is, and you can put a little brass ring around it, and then you can put hot stimuli or cold stimuli or chemical stimuli on top of that ending to see what it responds to. And when you do that, you get records like this. So these are recordings from a single axon, not a nerve bundle, but a single axon innervating the urinary bladder, in this case of the mouse. And each one of the vertical lines on this slide is an action potential recorded in that single axon. And so you can see that there are groups of fibers that innervate the bladder that we call muscular because they respond only to stretch or filling of the bladder, and they don't respond to stroking the urothelium. Then there's a group of fibers that respond to stroking of the urothelium, but they don't respond to stretch. And then there's also a group of fibers that respond to both of those stimuli. Now, there are other types of fibers that respond to only chemical stimuli and don't respond to mechanical stimuli. But the point of this is to show you that there are many different types of afferent endings in the bladder, and they respond to different types of stimuli. And that's important because when you begin to look at what's expressed in these nerve terminals, they're not all the same. They're different. So some respond only to low pH. Some respond to a mechanical stimulus. Some respond to an inflammatory mediator. And so it becomes difficult to say there's one factor that contributes to bladder pain or discomfort. The other thing to appreciate, and I tried to emphasize this earlier, that there are two nerves that innervate each of these organs. And so what you see here on the left is a picture of that bladder that's been splayed out. And these are receptive endings that we found in the bladder. You see that many of them are green, and they refer to neurons that responded to stretch or filling of the bladder in the pelvic nerve innervation. If you look at the lumbar sympathetic innervation, you see it's quite different. There are fewer neurons that respond to stretch, many of them that respond to manipulation or probing on the serosal surface of the bladder. I don't know what that's all about, but that's what they do. And you can see that they're quite different. So the receptive endings in the bladder are distributed differently by the two nerves, and they also have different response properties. Let's talk a minute about what sensitization really means. I showed you that leftward shift. Here's a leftward shift essentially in a single axon. So what sensitization means is that response magnitude increases, response threshold decreases, and sometimes spontaneous activity develops. Spontaneous activity in a nerve that's signaling information about discomfort or pain is not a good thing because it provides chronic input and suggests that there's painful stimuli. So here you're shown a neuron that responds to increasing intensities of stretch of the bladder. We then inflamed that particular area that had that receptive ending in the bladder and then tested it again, that same neuron. And you can see that response magnitude is increased significantly in this inflamed condition, and response magnitude has decreased significantly. If you look at the response in the control circumstance at 10 grams of stretch, and now the response at 5 grams of stretch, you can see that that's almost as great as 15 grams in the normal condition. So what does that mean? It means that if your bladder begins to fill and you have this interstitial cystitis or bladder pain syndrome, you're getting a signal from a low volume of urine in the bladder that's generating an incorrect interpretation in the central nervous system. And so you've got urge and you've got frequency, and that happens because the nerves are sensitized. They exhibit sensitization for a long time. So this is work from the mouse colon in which we had a model of irritable bowel syndrome and a model of colitis that we developed. We studied these animals, and then 3 to 4 weeks after the insult and after the inflammation had resolved, we recorded from these fibers. And I just want to show you that. You see that in the IBS-like model, the red line represents a leftward type shift at 25 days after treatment, and in the colitis model at 4 weeks after treatment, the green line shows again this leftward shift. There's no inflammation present. The colon looked entirely normal, but the sensitization persisted. So if this happens in a human condition, you can see that even if you do a biopsy and you don't see anything wrong, it doesn't mean that the nerves are normal. So afferent fiber sensitization initiated by organ insult can persist for a long time. So let me finish up here with a study that we did looking at the prostate and the influence of prostate inflammation on bladder function. So you see a cartoon here with the bladder and the prostate with a single neuron in the dorsal root ganglion and again with a cell body in the spinal cord. So you can take a dye, a tracer, and inject it into the bladder wall, and you can take a different color dye, a blue one, and inject it into the prostate. And these dyes are taken up by the nerve terminals in the bladder wall and in the prostate, and after 7 to 10 days or so, they're transported back to the cell body. And so then you can take out the dorsal root ganglion, and you can look at it under a microscope with a filter, and you can look at where the cells that have blue are located. And in the same slide, you can change the filter and look at the neurons that have the red dye in it. And what you see when you merge those images, you see a bunch of neurons that have both dyes in it. So this suggests, in fact, that a single cell body can have an axon that ends in 2 different organs. Now, this is a small proportion of the neurons that innervate the bladder, maybe only 10% or so. But still, they may play a role. It's not clear that they do at present. But certainly there's an effect of prostatitis on bladder function. So what we did is we then looked at the bladder response to distension. And you can see in the black line, saline-treated animals show a response like this. And in the inflamed prostatitis animal, the bladder response is exaggerated significantly. Again, a leftward shift, a sensitization. And if you look at neurons recorded in the pelvic nerve innervation of the urinary bladder in animals with prostatitis, you see that the response, again, is quite exaggerated in these inflamed animals. So there are certainly gaps in our knowledge. We know that the nervous system is important. I didn't talk about the sympathetic or the parasympathetic efferent role that may influence these sensory neurons. We don't know anything about how the sympathetic input to the bladder, for example, influences the sensory output. We know that the urothelium is involved and releases all kinds of things. Lori Berter and her colleagues have worked on that for quite a while. But we don't understand exactly what's happening at the interface of that sensory neuron in the bladder and some of these other chemicals that might be released from sympathetic or parasympathetic efferent nerves. That's a big mystery, so that's a gap that we have to fill. What are the molecules that initiate this sensitization? People have looked at tons of immunomodulatory molecules, growth factors. We don't know what the role of the urothelium is exactly, but it plays a role certainly. Cross-organ sensitization. Question is there a significant role for one neuron that innervates several organs? And I didn't talk about these descending influences that may influence, of course, the sensitization process in the spinal cord, because there are descending inhibitory and descending facilitatory influences that come from the brain and can dampen down or can release from control those neurons in the spinal cord. So I'm not suggesting the brain's not important. If you didn't have one, it would be a big problem. But you have to understand that studying that interaction is different than trying to understand a mechanism at a local level. So I'd like to thank you for your attention and let you know where I am in the summertime. Thank you.